Good morning. My name is Divyashree and I'm currently standing in the fish room that also belongs to our laboratory. And this is the room where we maintain and keep all our zebra fish. And so we feed the zebra fish 5 times a day. The feeding timings are from 9 o'clock to 10 o'clock, 12 to 12:30, 2 to 2:30, 5 o'clock to 5:30, and we also feed from 7 to 7:30. So the type of food we feed the fish are all different during different timings. At 9 to 9:30 in the morning, we feed them with fish food. And then between 12 to 12:30, we feed shrimp. And then again from 2 o'clock to 2:30, we feed them with fish food again. And then at 5 o'clock, we again give them shrimp. And at 7 o'clock, we again give them fish food. So we feed them with shrimp twice a day. And we feed the adult fish with this food. So for the larvae fish, the food is again different. So we keep the larvae on one side separately. So this section is only for the larvae. So for the larvae, as you can see, we have labeled each tank with different food labels. So this container is the larvae food. So when the larvae are 5 DPF or 5 days post fertilization or even 6 DPF, we transfer the larvae into these tanks, and these tanks are a little bit different because of the filter. So let me show you. So the larvae tanks usually have a very different small filter. That's the reason these tanks are a little bit different, and this filter prevents the larvae from moving into the circulation system or getting washed away. So the moment the larvae are transferred into these tanks, since they are very small, we first feed them with paramecium. So wherever you see a label that only has paramecium, you will know that the larvae has been transferred very recently. And as the larvae starts to grow, we attach labels. So when the larvae is a little bit more older, we attach the larvae food. And as the larvae grow a little bit more, we then attach the shrimp. And after they grow a little bit more, we move them to the adult side. So now I have feeding duty. So since the time is from 7 to 7:30, I am going to feed all the fish tanks. We have fish tanks arranged on this side and on the other side. So before we start the feeding, what we need to do is we need to stop. You can see there is water flow. So when we start feeding, we need to first press this button so that all the water flow has to stop. As you can see, it has stopped. Only once this stops, we may start the feeding. So when we start the feeding, we just open it and we tap it twice. If the tank has a few more fish, like a bigger tank, then we will feed them with more food. We must do this slowly because the fish might tend to jump outside. So we are going to do it a little bit slowly. Just for the sake of the video, I am going to feed the larvae, but I will continue to feed the rest of the fish. So I'm going to feed larvae food. So I can use the larvae food only for whichever are labeled larvae food. So I'm going to feed this one. And now I'm going to feed paramecium. So what we do is, for the paramecium, this is the paramecium culture. So we must, to avoid contamination, we do not touch, we keep this on top and we do not keep it on the bench. And what we do is we feed 50 ml per tank. Since this is empty, we will take some more. Usually if there is excess, we leave it so that the person who will feed next will continue to feed from here. And on this side, what you see over here is how we maintain the shrimp. So what in the fish, the shrimp preparation is usually done at 12 to 12:30. So the person who comes to feed shrimp at 12 to 12:30 also has the duty of preparing the shrimp. So over here we have first a brine solution. To prepare the brine solution, what we do is we have this salt. We measure 150 grams of this salt and then we mix it with tap water. And what we do is we turn on the motor. The motor is turned on and then we first put the salt in the salt. We take the salt, we put tap water, and now when the motor is on, it causes the salt to first dissolve. The salt is dissolved. After the salt is well dissolved, we take shrimp. We take shrimp eggs. Let me show you the shrimp eggs. So this is how the shrimp eggs look. This content has been emptied into this for easier use. So what we do is we take 2 full spoons of the shrimp eggs and then we add it into the brine solution. Now these eggs will take 24 hours to hatch. So if we are going to prepare the salt water, put these eggs, then we can use the shrimp only on the next day. So usually whenever the person comes for shrimp feeding, they are using the shrimp that has been collected at 9:30. So according to how things work is from 9 to 9:30, the first feeding, the person will feed fish food, but the collection of shrimp will happen. Collection of shrimp will happen at 9 o'clock. Preparation of shrimp will happen at 12. And then we also feed the paramecium culture I just used. So the paramecium culture is fed at 2 o'clock. For the paramecium, we feed them with yeast. There is again a certain procedure we follow while feeding the paramecium. We take yeast from, this is the yeast that we take. And for this, we have a separate equipment again. So everything in this is used only for the paramecium to again avoid contamination. What we do is we use this spoon to take half a spoon of the yeast is taken. We mix it, we put the yeast into this and then we fill it up to 20 ml of water, but we use only RO water. After filling it up to 20 ml, we thoroughly mix the yeast and then we follow what is written over here. So what for 1 liter we will use around for every liter we will use around 0.6 ml. So since there is around 12 liters here, since we will feed for 12 liters, it's around 7.2 ml. So after doing this, we will use a dropper and we will take the yeast solution and we will feed the paramecium. So this again happens. This happens at 2 o'clock. So like I said, and then after the person is done, we need to sign in this. So like you can see here, we feed fish food at 9 to 10. We collect the shrimp in the morning, same time 9 to 10. Shrimp is again fed at 12 to 12:30. We prepare the shrimp by the salt water method. Again, we feed fish food at 2 o'clock and we feed the paramecium at 2 o'clock. These two events happen together. And at 5 o'clock to 5:30, we feed shrimp. And 7 to 7:30, we feed fish food. So this and the numbers you can see here, shrimp hatchery. So if the shrimp is, so this number 1 indicates that today's date is the 8th of June. So today the person who collected shrimp, he collected it from 1. So since he collected it from 1, tomorrow when we prepare shrimp, or the one that we will be using will be 2. So we alternately choose, so like I said, it's labeled as 1 and we label it as 2. So according to the, if today we have used 1, then tomorrow we will be collecting the shrimp. Shrimp collection will take place from this one. So when we collect shrimp again, there is a certain procedure that we follow. I will show the shrimp collection in another video. Okay, so over here you can see that we have set up mating. So there is a difference between the mating tanks and the normal tanks. The normal tanks, like you can see, they do not have any separation or any blocker in the middle. And the lid of the normal tanks are usually blue in color. But the fish that have been, these kind of tanks is what we call a mating tank. And I will show you the mating tank in more detail. So this is the kind of tank we use. And this is placed inside, we place this inside this. You may notice that the bottom of this tank is a bit porous, and this is porous because when the fish lay the embryos, we want the embryos to pass through this and get collected at the bottom of this. So what we do is, it's usually like this, and we fill it with water up to here. The females are put on one side and the male fish are put on the other side. So what we do is we set up for mating on the previous day. In the sense, if I want embryos tomorrow morning, then I should set up for mating today evening. And we separate. So usually the standard number of fish we use are 4 females and 2 males. You can also use 8 females and 4 males. It depends on the kind of cross. So the crosses that I have been performing are self crosses, but it depends on the number of embryos, and there are many factors that determine how many males and females you are choosing. So once you fill the tank with water, we put the males and the females, and then we cover this, and then we place it overnight. And then next day in the morning, as early as possible, preferably around 9 a.m. or before 9 a.m., we come and we remove, this is called a blocker. So what we do is we come at 9 o'clock, we come in the morning and we remove the blocker. So the moment we remove the blocker, the fish start mating with each other, and we allow the fish to mate for 2 time periods. One is 20 minutes and one is 40 minutes. So if we get the desired number of embryos in the first 20 minutes, then we need not allow the fish to mate for 40 minutes. But some of the fish, since they take more time for mating or laying embryos, we allow them to mate for 40 minutes. So when we remove the blocker and when we allow them to mate, we try not to scare the fish and be in the fish room. So after removing the blocker, we step outside and give the fish some time and privacy to mate. And after that, we come and we collect the embryos. And after the mating, the fish are put back into the normal tank again, and then they're put back. Another thing we do before mating is, in the night, whatever water we fill is up to this level. But in the morning, what we do is we fill minimum water, because the fish should not be, it is also to allow the embryos to be in one area, and more water will provide the fish more area. So to increase the chances of mating, we try to keep the fish in lesser water. So this is the lid that we use to close the tank. So this is the rack where we arrange all the mating tanks. So this person has set up 6 tanks, 6 mating tanks. Another thing about the fish room is we arrange the fish according to the type of genotype or the strain. So this area over here is only for the wild type fish. So the way we set up a wild type, okay, so whenever we are conducting an experiment, I conducted an experiment using mutant fish. So as a control we breed wild type fish, because this will behave as a normal reference or a standard to which we will compare our mutated fish. Wild type, the way we set up mating in our lab, we do not we do it in a very cyclic way. In the sense, if I set up wild type mating with this tank, I will make note of it, and the next person who sets up mating will have to use the next tag. In this way, we give enough time for the fish to recover from mating, because the female fish cannot keep laying eggs. So whoever uses wild type fish, we use it one after the other, and we give enough time for so the way we use it, it follows the numbers. So over here you will see 1, 2, 3, 4. So it goes on up to 41 to 42 to 44. So once this tank has been used, finally we come back again to using the first tank. That's how this goes. And my mutant fish, POMGNT1 fish, are placed on the other side. So I also have a few larvae fish. So my younger fish are placed on this side, and my other adult fish are placed on the other side. So according to the strain, we have arranged fish differently. We also have a few unique zebra fish in our lab. The fish are not mine, they belong to another person, but I will just show the fish in the video. So these fish, like you can see, they are pink in color. So the strain of the fish is a little bit different over here. So the way we label our fish is the date of breeding, the day, just like how I described the mating process. So when we describe the mating process, the day we collect the embryos, we call it, just like how we call DOB or date of birth, like that we call it date of breeding. So that is a breeding date. So usually in the label, we mention the strain, then we mention the date of breeding, and then the owner's name is mentioned. Our lab number is also mentioned, and then this code is also the ISUC code. So this is how every single tank is labeled, and we also have a nice circulation system where the water, each tank has an individual supply of water, but the fish do not require any oxygen, because now they are capable of using, they have the ability to breathe without requiring dissolved oxygen. So only the larvae fish require external oxygen tube. And on this side, I also have my fish, the fish that I maintain. So all these tanks belong to me. As you can see, I've mentioned my name, I've mentioned the gene I'm working with is POMGNT1, but then this fish are homozygous. So that's why it is 12-188. This is the homozygous variant. And these fish are heterozygous. So according to my experiment, I've used A and B. So for that convenience, we label them with A, B and C. But I just wanted to show where I keep my fish in the lab. Thank you.